Hello, everyone, and welcome to the first International Electronic Conference on Toxins. This conference has been organized by MDPI and the Toxins Journal. I would like to thank our chairs and the conference committee for making this conference possible and all the participants for joining us today. I have some things to remind everyone. First of all, this conference is now open for discussions. So that means that until January 31, you can read and comment all submissions presented to the conference. Second is that tomorrow we will also have a live session. So please check the conference website for information and to register for free. And third is that this session is being recorded and that the recording will be posted in the conference webpage for everyone to watch it as soon as possible. Our session today will be focused on unique approaches to tackle snakebite-induced deaths and disabilities. And our session chair will be Dr. Vaiyapuri from the University of Reading in UK. We will have three main speakers, including Dr. Vaiyapuri, Professor Dr. José María Gutiérrez from Instituto Clodomiro Picado from the University of Costa Rica, and Dr. Andreas Hougaard Laustsen from the Technical University of Denmark. After each presentation, we will hold a question and answer session. We will address any question that you submitted during the session. So if you are joining us live, please type your question in the QA box at any time. Finally, I'd like to introduce our session chair, Dr. Sakthivel Vaiyapuri, who is an associate professor of cardiovascular and venom pharmacology at the University of Reading, UK. He's also a Royal Society Leverhulme Trust Senior Research Fellow for his research on snakebite-induced muscle damage. His research is mainly focused on establishing the pharmacological actions of venom toxins in inducing various envenomation effects. The research in his groups aims at understanding the differential effects of venoms on skeletal muscle and the impact of next-generation antivenoms in preventing and treating snakebite-induced muscle damage. Thank you very much, Doctor, for accepting to chair the session. The microphone is yours.
Excellent. Thank you, Anna. So, good afternoon, everyone, or good morning if you're joining from Western countries. It's my great pleasure to welcome you all for this wonderful webinar on snakebites through this International Electronic Conference on Toxins. So, as Anna introduced, I'm Sakthivel Vaiyapuri from University of Reading. I'll be chairing this session. And we got two other fantastic speakers for this session. So today we are going to talk about unique approaches to tackle snake bites. We are calling this as a unique approach because this webinar contains unique approaches. So the first speaker is our Professor José María Gutiérrez. He's going to talk about snakebite-induced muscle damage, particularly local envenomation effects. So this is an under-researched area where José María has spent a lot of time researching on this particular area. And this is always a challenge for clinicians to treat or prevent snakebite-induced muscle damage or local envenomation effects. So this is an important area that we all should understand in order to better treat or prevent snakebite-induced damage. Our second talk is going to be on modern recombinant antivenoms, humanized antivenom, and that's a toxin specific. We all heard about polyvalent antivenom for centuries, and we know that they are not perfect, although they are good enough to save lives. But now we need to move towards the next generation antivenoms. So this talk is going to be given by Dr. Andreas Laustsen from Technical University of Denmark. So Andreas has done a lot of work on developing toxin-specific antibodies, specifically this recombinant one. So I will also introduce Andreas later on. So finally, I'm going to talk about community health education for snakebite, because despite all the advances we do for research and development, if people don't bother seeking prompt treatment, or people don't develop their own training skills in order to handle snakebite victims in the local healthcare settings, then this is going to be a problem. So I'm going to talk about the program we've been running in India for the last 2 years on community public health education. So I hope you will all enjoy all these three talks. And as Anna mentioned, please feel free to post your question in the Q&A chat box, and we will try to answer questions after each talk as well. So without further delay, I would like to introduce Professor Jose Maria Gutierrez. All of you would have heard about Jose Maria. He's a legend in snakebite field, and he has published more than 250 papers. So if you are doing venom research, you won't do research without Jose Maria's papers. So currently, Jose Maria is an emeritus professor in the University of Costa Rica. And despite all the other research, Jose Maria spent a lot of time on local envenomation effects. And you can look at the literature, there are only very few people who are researching on local envenomation effect, and Jose Maria is the leader in that field. And he has discovered a lot of new things about biotoxic phospholipase A2 and the metalloproteinases and the mechanism behind the local envenomation effects. So in addition, Jose Maria has developed antivenoms to treat actual snakebite cases in several countries, particularly in developing countries. And he's been raising awareness about snake bites in Latin America. And Jose Maria is also a member of board of directors for Global Snakebite Initiative. So it's my great pleasure to welcome Professor Jose Maria to present his talk on snake venom hemorrhagic metalloproteinases. Jose Maria, it's yours.
Thank you very much. Good morning or good afternoon, depending on where you are. I'd like to thank Dr. Vaiyapuri for his kind invitation to be at this symposium. And it's my pleasure to join this activity with Dr. Vaiyapuri and my colleague and friend, Andreas Laustsen. So I will share my screen now. Greetings to everybody and to the people that are watching this activity. I'm going to talk today about the mechanism of action of snake venom hemorrhagic metalloproteinases. It is well known that local and systemic hemorrhage are very characteristic features of snakebite envenoming, especially by viperid snakes, by snake bites. This is manifested in terms of local hemorrhage, which involves tissue necrosis and also impairs muscle regeneration, and also systemically by hemorrhage in different organs and tissues, which may lead to a cerebrovascular accident and pulmonary hemorrhage, as shown in this light micrograph of lung tissue from a mouse injected with a hemorrhagic venom, showing many erythrocytes in the alveolar spaces. So local and systemic hemorrhage are typical and very important features in the snakebite envenomings by viperid snakes all over the world. This activity, the damage to microvasculature and the hemorrhagic activity, is due to the action of zinc-dependent metalloproteinases in these venoms. And these are very abundant in viperid venoms. Here I show the case of the snake Bothriecis lateralis, a viperid from Central America. And in the proteome of this, the venom of this snake, metalloproteinases comprise more than 50% of the venom. And this is the case in many viperid venoms in the world. Metalloproteinases not only induce hemorrhage, they also affect the coagulation and other physiological processes. But I will focus today on the development of hemorrhage. These metalloproteinases are a multi-domain family of enzymes in the venoms, and they have been classified in 3 different classes. The class P1 metalloproteinase is comprised by enzymes containing a metalloproteinase domain only. And in this metalloproteinase domain, you have the catalytic site where the zinc ion is present, which is absolutely necessary for the hydrolysis of proteins. Then there is another class called P2 metalloproteinases, which in addition to the metalloproteinase domain, have a disintegrin domain. And usually this disintegrin domain is cleaved once the protein is synthesized, and disintegrins are released. Disintegrins by themselves exert a number of action, particularly inhibition of platelet aggregation. And then we have the P3 class of metalloproteinases with several subclasses. And this class of metalloproteinases contain In addition to the metalloproteinase domain, a disintegrin-like domain, and then a cysteine-rich domain with a hypervariable region at the C-terminus of the molecule. And this can be either monomeric or dimeric, or in some cases, this metalloproteinase subunit is linked to two additional subunits, C-type lectin-like components, which allow these toxins to bind to specific targets, particularly to clotting factors. In general, P3 metalloproteinases are more hemorrhagic, are more active as hemorrhagic toxins than P1 metalloproteinases, and we will discuss why this is the case in this talk. If we see the 3D structures of P1 and P3 metalloproteinases, we see that in the P1, we have the metalloproteinase domain with the catalytic site here. You see the zinc ion and 3 histidines which coordinate this zinc ion. There is also a glutamate here. And very close to this active site, there is a loop which we will see that plays a key role in defining whether a P1 metalloproteinase can induce hemorrhage or not. In the case of P3 enzymes, you see the metalloproteinase domain, but then in a sort of C-shaped structure, you have the disintegrin-like domain followed by the cysteine-rich domain. So please keep in mind that P3 metalloproteinases are more active as hemorrhagic toxins than P1 metalloproteinases, and snake venoms have different combinations of P1 and P3 metalloproteinases depending on the particular venom. The studies on the mechanism of action of hemorrhagic toxins started in the decade of 1970, particularly by the work of Akira Osaka in Japan and his group. They use electron microscopy and other methodologies to analyze the mechanism of action of these venoms and hemorrhagic toxins, particularly from the venom of the habu, Protobotrus flavoviridis. What they observed was that erythrocytes escaped the vasculature by a mechanism named diapedesis, which means that erythrocytes escape through widened intercellular junctions between endothelial cells, as observed here and here. It was observed that this type of escape from the vasculature by erythrocytes occurred mostly in venules. Later on, the work by Charlotte Ombie and other groups and colleagues working mostly in capillary vessels described a different type of mechanism for hemorrhage. And this mechanism has been called hemorrhage per rhexis. When the capillaries are observed in tissues affected by hemorrhage, the observation is that erythrocytes escape not through widened intercellular junctions, but through disrupted endothelial cells. So the cells become very thin in the capillaries, and then there is a disruption in the integrity of the wall where erythrocytes live, as shown here in the case of a metalloproteinase by Crotalus atrox, or here in the case of a metalloproteinase from Bothros asper. So these are two different mechanisms for extravasation, and we believe the difference between them basically lies in the type of microvessel being observed. So diapedesis occurs mostly in venules, and rhexis occurs mostly in capillaries. We believe that the most important mechanism is rhexis because hemorrhagic activity occurs basically at the capillary bed of the microvasculature. So if we want to understand the mechanism of action of hemorrhagic toxins, we need to study what goes on with the structure of the capillary when they get in contact with metalloproteinases. And just to review, the structure of the capillary is based on the presence of an endothelial cell, which is characterized by the presence of many vesicles called transitotic vesicles. And this endothelial cell is surrounded by a layer of extracellular matrix known as basement membrane. Basement membrane is very important for the capillary because it provides mechanical strength, mechanical support to the capillary. It is a scaffold for the capillary. And then you see the intercellular junction between endothelial cells in the capillary. So this is the locus where hemorrhagic metalloproteinases act. And we have been, and other groups have been studying how these metalloproteinases disrupt the integrity of capillary vessels, which we will review in the next slides. So the first question is, what goes on with the endothelial cell? Because in vivo, as we saw in the electron micrographs, there is a very rapid disruption of the integrity of endothelial cells in terms of minutes. I mean, you can see disrupted endothelial cell 5 minutes after injection of hemorrhagic toxins. So to understand the action of these toxins on endothelial cells, we and other groups have analyzed the action of these toxins on monolayers of endothelial cells. And this is a monolayer of endothelial cell. There are several types of endothelial cells that can be studied. When you apply a metalloproteinase into these cell layers, what you observe is a detachment of the cells from the substrate, owing to the action or the proteolytic action of these enzymes in the substrate. But when you analyze whether these cells are disrupted, are dead or not, by measuring the release of lactic dehydrogenase, which is a cytosolic enzyme that is released when the plasma membrane is disrupted, it is interesting to note that metalloproteinases do not induce a release in lactic dehydrogenase from these cells. In contrast, if you apply a myotoxin, which is a cytotoxin, a toxin that disrupts the integrity of plasma membrane, you see an increase in the medium of lactic dehydrogenase. So very clearly, hemorrhagic toxins applied to endothelial cells in culture are not directly cytotoxic. This detachment of the cells as a consequence of the proteolysis of the substrate eventually results in the development of apoptosis through a mechanism known as anoikis, which depends on the lack of contact of endothelial cells with their substrate. But this occurs hours after the administration of the toxin. So there seems to be a difference in the action of metalloproteinases in endothelial cells in vivo versus in vitro. And this was an intriguing finding. The next locus of activity that we explored was the action of metalloproteinases on the basement membrane that surrounds the endothelial cells. And this cartoon depicts the complexity of the basement membrane, which is comprised by a number of proteins such as nitrogen, perlecan, which is a proteoglycan, laminin, and type 4 collagen, forming a meshwork of proteins that provides mechanical strength to the basement membrane. So since the decade of 1980, several groups, initially the group of J. Fox and John Jarnason in the United States, and also the group of Osaka in Japan and other groups like ours, have explored the action of metalloproteinases in the basement membrane. And for this, it has been very useful to use a preparation of basement membrane known as matrigel, which is obtained from a sarcoma. And in vitro, you can study the action of metalloproteinases in matrigel. And this is usually done by incubating matrigel with the metalloproteinases and then running SDS electrophoresis to see whether there is degradation of the matrigel components. You see here in the control several bands of matrigel corresponding to laminin, to nitrogen, and so on. When this matrigel is incubated with metalloproteinases such as a P3 and a P1, you see the appearance of new bands which correspond to degradation bands, showing that metalloproteinases are able to degrade the components of the basement membrane. So one issue that we found in our first studies was this contrast between a rapid disruptive action of metalloproteinases in endothelial cells in vivo versus the lack of action of these metalloproteinases in vitro, except for the detachment of these cells. So that was an intriguing finding. And we wonder what was the basis for these conflicting observations between in vivo and in vitro experiments. And at that time, we came out with this idea that biophysical factors operating in the circulation would explain that this difference. And which are these biophysical factors? So the one important force working in the hemodynamics of microcirculation is the wall tension. And the wall tension in a vessel is directly proportional to the transmural pressure. That means the hydrostatic pressure of blood and also of the radius of the vesicle, and is inversely proportional to the wall thickness. So the tension in the capillary wall is determined basically by the hydrostatic pressure of blood. And another important biophysical factor in circulation in vivo is the so-called shear stress, which is a tangential force, which is directly proportional to the blood flow and to the blood viscosity and inversely proportional to the vessel radius. So we think that you need to incorporate this component, this element of biophysical factors for understanding the mechanism of action of toxins. And for this, we developed this model, a so-called two-step model to explain the mechanism of action of hemorrhagic toxins. The first step would be basically the cleavage of the basement membrane components as shown in vitro by the action of these metalloproteinases. This cleavage would result, and also the cleavage of proteins involving cell matrix interactions. This cleavage would result in the reduction in capillary wall resistance to distension, because as I mentioned, the basement membrane determines the mechanical stability of the capillary. So with this weakened structure in the capillary, the biophysical forces just discussed cause the distension of endothelial cell, which is made possible because of the weakening in the mechanical strength of the basement membrane. And this distension results in the thinning of the endothelial cell, the loss of pinocytotic vesicles, and eventually the disruption in the structure of the endothelial cell and the endothelium, and the extravasation of blood. So these are two necessary steps of different sort. The first one is a biochemical degradation of the proteins, and the second one is a physical distension and disruption in the integrity of the microvessel. To test this hypothesis, we predicted that in conditions in vivo where blood flow is interrupted, there wouldn't be disruption on the endothelial cell. And we checked this hypothesis in an electron microscopic study. And here I show an electron micrograph of a capillary vessel from a tissue injected with saline solution, showing the normal structure of the vessel with the pinocytotic vesicles and so on. Then in tissue injected with BAP1, a hemorrhagic metalloproteinase, in conditions of normal blood flow, you see the thinning of the vessel, the distension of the vessel, and the disruption of the wall, as described. But then, interestingly, when we injected this toxin in a muscle without blood flow, in conditions where the blood flow was interrupted, there is degradation of the basement membrane, as shown by Western blot and other techniques, but there is no disruption of the endothelial cell. And we believe this is so because there are no biophysical forces operating here that would distend this endothelial cell. So these observations led support to the two-step mechanism of hemorrhagic toxins. Another intriguing finding was that both hemorrhagic and non-hemorrhagic metalloproteinases are able to hydrolyze basement membrane components. And let me explain that not all metalloproteinases are hemorrhagic. There are many metalloproteinases, particularly P1 metalloproteinases, which are not hemorrhagic, and yet they are able to degrade basement membrane components. So how can this be explained? And this question brings another question. Which are the key targets of metalloproteinases whose hydrolysis affects the mechanical stability of the basement membrane? So we went on to analyze which components of the metalloproteinases were degraded. And for this, we compared with the collaboration of Eladio Flores Sanchez from Fundação Ezequiel Diaz in Brazil, we compared a non-hemorrhagic metalloproteinase P1 and a hemorrhagic metalloproteinase to see whether they would cleave in a different way the proteins of the basement membrane. And for this, what we did was to inject the metalloproteinases in the muscle of mice, then to collect the muscle, prepare muscle homogenates, and identify the degradation products by using antibodies against each of the basement membrane components by Western blot. And this is reflected in this slide. Basically, we use antibodies against laminin, nidogen, type 4 collagen, and perlecan. And it is basically we run the homogenized muscle of a control injected with saline in all the experiments, and then from muscle injected with a non-hemorrhagic P1 and a hemorrhagic P1 metalloproteinase. You see that there is no difference in the pattern of degradation of laminin between these enzymes and of nidogen. But when we examine the degradation of type 4 collagen, we found that the non-hemorrhagic enzyme practically did not degrade the type 4 collagen, where the hemorrhagic one caused a significant degradation of type 4 collagen. And also we found differences in the degradation of perlecan, which is a heparan sulfate proteoglycan. These two components play key roles in the mechanical stability and structure of the basement membrane. So owing to the role of type 4 collagen in determining the stability of the basement membrane and the overall assembly of the basement membrane, we hypothesize that the cleavage of type 4 collagen is a determinant factor for the ability of these toxins to induce hemorrhage. Type 4 collagen is a protein, a component formed by a triple helical structure. But the most important thing here is to know that both at the N terminus, at the C terminus, they have covalent bonds which link different proteins or different molecules of type 4 collagen, therefore providing enough strength and mechanical stability to the basement membrane. So it makes sense that cleaving this component would reduce the overall mechanical strength of the basement membrane. Also, in collaboration with Jay Fox at the University of Virginia, we analyzed by proteomic analysis, the composition of the exudates collected from tissue injected with hemorrhagic and non-hemorrhagic P1 metalloproteinases. And I'd like to remark from these observations that exudates collected from hemorrhagic, from tissue injected with hemorrhagic toxins have much higher amounts of a basement membrane heparan sulfide proteoglycan core protein fragments, therefore corroborating that the hemorrhagic one degrades proteoglycan to a higher extent. But we also observed a novel finding in that there was also a higher amount of fragments from another type of collagen, type 6 collagen. And type 6 collagen is important because it bridges the basement membrane with the interstitial fibrillar collagen meshwork in the extracellular matrix. So if we see this cartoon depicting the structure of the basement membrane and the nearby fibrillar extracellular matrix components, we see that type 4 collagen and perlecan are key components of the basement membrane, and therefore their cleavage by hemorrhagic metalloproteinases results in the destabilization of the basement membrane. But also collagen type 6 bridges the basement membrane components with the fibrillar matrix of the extracellular matrix. And this, the disruption of type 6 collagen could be also relevant for the overall weakening of the stability of this extracellular matrix structure. So it is interesting to analyze why, in terms of structural determinants, is one P1 metalloproteinase able to induce hemorrhage, whereas others are not. Since the overall 3D structure of these P1 metalloproteinases is very similar. It's almost superimposable. So comparing these structures, it has been noticed that the main difference between hemorrhagic and non-hemorrhagic ones lies in this loop located close to the catalytic site. And this is a closer look of this structure of this loop, which is very close to the catalytic site and may affect the ability of the enzymes to induce hemorrhage. And in a molecular dynamic study done by Wellnerfer and colleagues, they analyzed the dynamics, the flexibility of this loop in hemorrhagic and non-hemorrhagic metalloproteinases. And it was interesting to note that the hemorrhagic ones show higher flexibility in the first part of the loop, whereas the non-hemorrhagic ones showed higher flexibility in the latter part of the group. So it may be that the sequence of residues and the flexibility that comes as a consequence of this sequence may determine whether this loop affects or binds to type 4 collagen and other components of the matrix and enabling the toxin to degrade these components. To test this hypothesis of the role of these residues in different parts of the loop, Erika Camacho from our group prepared recombinant versions of VAP1, a P1 metalloproteinase, which is hemorrhagic. And in one of these recombinants, she mutated the residues in the first part of the loop and introduced residues corresponding to the non-hemorrhagic metalloproteinase, and then tested whether this affected the hemorrhagic activity. And as shown in these pictures, with depict the hemorrhagic lesion in the skin of mice, the recombinant BAP1 induced the typical hemorrhagic spot characteristic of hemorrhagic metalloproteinases, and the mutant one lacked hemorrhagic activity, therefore supporting this hypothesis that the residues comprising this first and second part of the loop may determine the ability of these toxins to bind, interact, and degrade extracellular matrix components. Then we went on to ask whether this finding of degradation of type 4 collagen was common not only in P1 metalloproteinases, but it was also observed in P2 and P3 metalloproteinases. And for that, we injected P1, P2, and P3 metalloproteinases from snake venoms into mice, collected the tissue, prepared homogenates, and analyzed the degradation patterns by using antibodies, as I previously described. And it was interesting to see that the pattern of degradation of type 6 collagen, laminin, and nitrogen were different depending of the enzyme. And I must say that to do this experiment, we injected an amount of enzyme that induced the same extent of hemorrhagic lesion. So the prediction was that the protein that would play a key role in this mechanism of action would have the same pattern of degradation in the three toxins. And the only component that showed the same pattern of degradation in these conditions was type 4 collagen, thus corroborating that also P2 and P3 metalloproteinases are also able to degrade type 4 collagen to the same extent. And this reinforces the idea that degradation of type 4 collagen is key to understand the mechanism of action of these proteinases. Now, why are P3 metalloproteinases generally more hemorrhagic than P1 metalloproteinases? And this may be related to the presence of exocytes in the additional disintegrin-like and cysteine-rich domains, which may allow these toxins to bind, to be targeted to particular sites in the microvasculature. And to test that, Cristina Herrera from our group, working in the laboratory of Susan Nurshak in London, analyzed in a model of mouse cremaster muscle in intravital microscopy the binding of P1, P2, and P3 metalloproteinases to the microvasculature. You see here, this is a microvasculature stained with type 4 collagen antibodies. And then here, the different toxins, P1, P2, and P3, added to the microvasculature to the preparation with a immunofluorescent label. And you see very clearly that the P2 and the P3 actually bind very specifically to the vessels, whereas the P1 has a more widespread distribution, owing probably to the lack of exercise that direct these other toxins to the vessels. So this may be the reason why P3 metalloproteinases are more active as hemorrhagic toxins, because they are directed to the microvasculature. And which are these exocytes? This is not known yet. Several groups in Brazil, particularly in Instituto Butantan, and in our group and other groups, have suggested that exocytes are located in the disintegrin-like domain or the cysteine-rich domain, but this has not been conclusively demonstrated so far. Finally, I would like to mention that all this knowledge can be applied to eventual new treatments for snakebite envenoming, and particularly the search for synthetic inhibitors of metalloproteinases is a very fruitful field of research nowadays. And some of these inhibitors are synthetic inhibitors that have been developed for other diseases such as cancer, inflammatory diseases, and so on. One example is batimastat, which is an peptidomimetic hydroxamate able to bind to the catalytic site of metalloproteinases and to chelate the zinc ion. And we tested this inhibitor for the inhibition of hemorrhagic activity by a very important snake in Africa, Echis ocellatus. And briefly, this shows that if the inhibitor is applied immediately after venom injection in the muscle of tissue, there is a drastic reduction in the extent of hemorrhage, therefore demonstrated that these inhibitors are effective and could be applied in the field in the treatment of snakebite envenoming. Finally, I'd like to thank my colleagues from Instituto Clodomiro Picado, Jay Fox, Michael Ovadia, and Gadi Borko, Susan Nurchar, and Mathieu Benoit Besson, Eladio Flores Sanchez from Brazil, and Torsten Lingot from Germany, and the economic support by University of Costa Rica, the Wellcome Trust, and the International Foundation for Science. And I thank you all very much for your invitation and your attention. Thank you very much.
Excellent. Thank you so much for this brilliant talk, José María. It's amazing to cover different types of metalloproteases and their actions. So participants, you can post your questions in the question and answer chat box. So there's one question in the chat box from Stephen Hall. So this is about LDH assay. They tried using LDH assay with Echis ocellatus venom, and apparently the venom is directly reacting with the reagents in the absence of any cells. So they are asking whether you encountered this type of issues before.
Mm-hmm. Yeah, this may happen because of the complexity of the venoms that may affect some of the reagents of the assay. The experiment I described were done with purified metalloproteinases. In that case, there is probably no such interference. But with working with crude venoms, this may happen. So you may try other ways to detect cytotoxicity, such as the MTT assay, for example, or the binding of propydium iodine to the cells using fluorescence. So my recommendation would be to search for other types of assays to measure cell disruption, and there are several assays available in the literature. Mm-hmm.
So there's another question from Maria Elena. Are the serum effective against these effects of SPMPs? I don't
Would you
think I
repeat that,
understand
please?
the question. Or the serum effective against these effects of SPMPs?
The serum.
Yeah, serum.
Yeah, yes. Well, depending on the species of animal, there are very potent inhibitors of metalloproteinases in the blood. Well, in fact, even in humans, it is known that alpha-2 macroglobulin, a generalized proteinase inhibitor, is effective. And this is interesting because it has been shown that alpha-2 macroglobulin is effective against P1 metalloproteinases, but somehow P3 metalloproteinases escape the inhibition of alpha-2 macroglobulin. And this may be another explanation why P3 metalloproteinases are more active systemically, especially than P1 metalloproteinases. But so this is an interesting observation. But also in the serum of several animals, like the opossum, for instance, the group in Instituto Osvaldo Cruz in Brazil has investigated the proteins in the opossum serum, which enables this animal to be resistant to the metalloproteinases. And these are very potent inhibitors of metalloproteinases. And there are other inhibitors in the blood serum of other species. So this is an interesting example of innate immunity to snake venoms. But the difference between P1 and P3 in terms of inhibition by alpha-2 macroglobulin is really interesting.
Yes, I would like to ask a couple of questions. I think we haven't got many questions. It's coming up now. So have you witnessed that in the disseminated intravascular coagulation after you inject your metalloproteases or whole venoms into the local tissues?
You mean if there are alterations in the coagulation?
Yeah.
Yes, well, there are several things here. If you work with crude venoms in viperic venom usually affect the coagulation. They have procoagulant components that induce coagulopathy, induce consumption coagulopathy, and therefore induce defibrinogenation, incoagulability. So this synergizes with the action of hemorrhagic toxins. Therefore, hemorrhagic toxins disrupt the integrity of the vessels, and these coagulopathies contribute to bleeding because the hemostasis is inhibited. The same thing with toxins that inhibit the platelet function or induce reduction in platelet numbers. So there is a synergism between the toxins that induce coagulopathy. But then, in terms of metalloproteinases, there are metalloproteinases, particularly P3 metalloproteinases, that also activate coagulation factors. There are factor 10 activators, prothrombin activators, and so on, that induce this consumption coagulopathy. But there is a difference between the hemorrhagic metalloproteinases and the procoagulant metalloproteinases. And using these cremaster muscle preparations, we have shown, Cristina Herrera has shown recently that, for instance, procoagulant P3 metalloproteinases do not bind to the microvasculature. So there may be differences in the exosites of P3 coagulant and hemorrhagic metalloproteinases. And also some metalloproteinases are fibrinogenolytic, so they degrade fibrinogen and therefore also may contribute to the coagulopathy. So this is a very complex scenario where different metalloproteinases exert different activities.
So two more questions in the box. So first one from Spiros Platches. Do you think that the differential effects of the toxins in vivo and in vitro are only related to the difference in biophysical factors? Or could there also be a difference in overall activity of the metalloproteinases and substrate targeting differ under different conditions?
Yes, I think this is a very good question because the two models used to explore hemorrhagic activity in vitro and in vivo are very different. And we do not know whether the endothelial cells used in cell culture experiments actually reproduce the phenotypes of endothelial cells in vivo. And this is why it's so important to bring new models, new in vitro models, to investigate the action of these toxins in the microvasculature. For instance, organoid type of models of organization of endothelial cells in three-dimensional structures in culture, which would simulate more closely the structure of the vasculature in vivo. I think we need to develop novel models, which some of them have been already developed, to test the action of metalloproteinases in vitro in models that resemble more closely the conditions of in vivo experiments. So these observations that I presented are initial approximation to this phenomenon, but there may be a possibility of reproducing in vitro the action of these metalloproteinases, perhaps introducing blood flow or guaranteeing that the phenotype of these endothelial cells would more closely resemble the phenotype of these cells in vivo. This is a very good point.
Yeah, the last question. What would be the best route of administration for drugs to intervene with SVMP-induced muscle damage or local inflammation effects? Because the capillaries will be already damaged. So
Yes. Well, this is also a relevant issue in terms of designing eventual future treatments for snakebite envenoming. The idea that we and other groups are developing is the possibility of administering these inhibitors in the field rapidly after the bite of the snake. So if inhibitors like batimastat or these inhibitors, which are safe, can be applied in the field, eventually, of course, after clinical trials that support this application, then these inhibitors would block the toxins before they initiate the disruption of the tissue. So the time frame here is very important. The longer it takes to administer inhibitors of antivenom, the more difficult is the neutralization, because the tissue is being disrupted rapidly after the injection of the venom. So the ideal scenario would be the possibility of injecting these inhibitors in the field rapidly after venom injection, and I would say injecting them locally. Then, of course, there is a possibility of administering these inhibitors systemically through the IV route, for instance, either in hospitals or in a health facility, that would also neutralize the circulating hemorrhagic toxins in this case, in addition, every time, to antivenom, which is able to neutralize all other toxins in the venom. But this is a growing field of investigation. There is very encouraging results regarding the use of metalloproteinase, phospholipase, and other inhibitors. And probably the future in snakebite envenoming therapy will be a combination of application of these inhibitors in the field and application of traditional or novel type of antivenoms in the hospital.
excellent. Thank you so much, Jose Maria. That's a brilliant talk and nice answers for all the questions. Thank you
Thank
so
you,
much
José,
again.
very much.
Now we would like to move to our next talk on broad spectrum recombinant antivenoms. So this is going to be delivered by Dr. Andreas Laustsen from Technical University of Denmark. So Andreas is a best friend of mine. Also, he's a young superstar in snakebite field. Andreas is a fellow of Young Academy of Denmark and also a fellow of Young Academy of Europe. And he is a co-founder for several startup biotech companies, many of them particularly focusing on snake bites for diagnostics and also therapeutics. And notably, Andreas developed the world's first fully humanized toxin-specific recombinant antibodies. And he's leading large research group with more than 20 people. And he won lots of big grants, including from European Union and also from Wellcome Trust. And now Andreas is going to talk about broad spectrum toxin specific recombinant humanized antibodies for snakebite treatment. Andreas, it's yours.
Thank you very much, Sakthi, and thank you so much for inviting me to give this this presentation. And also thanks to the Toxins journals for arranging this this webinar, which I think is a great way to get a lot of people from around the world gathered these days. So wait a second. So just a very brief introduction. As Sakthi said, I run a lab that's growing quite in numbers. We expect to be 35 scientists by summer. And we work a lot within toxinology, but have a particular focus on antibody discovery, of finding antibodies that can target not just snake toxins, but also toxins from other venomous animals and even bacteria. So I want to start this webinar a bit with a bit of a personal story. So the way I got into snakebite research started back in Tanzania in 2011, where I was on safari with my family. So we were just in Tanzania to be on vacation, but on this trip, we came across a snakebite clinic where we met two young children that had been bitten by spitting cobras. And I warn some of you that are not used to seeing nasty clinical pictures that the next 3 slides will show some quite nasty pictures of snakebite victims. So this is not the particular victim that we came across, but a similar type of case. So the two victims we saw were two children, age about 5 and about 8, that had been bitten by spitting cobras, like this child on the picture. So spitting cobra venom contains cytotoxins that go in and destroy tissue simply by disrupting cellular membranes. And not only will this cause a lot of local tissue damage, it can very easily lead to more systemic effects like sepsis if the wounds get infected. So that's an example of one of the, you could say, classical symptoms of a snake bite. Another typical symptom for neurotoxic snakes is flaccid paralysis. And the child in this photo has also received a cobra bite and is paralyzed. And if these neurotoxic effects, they progress, this can lead to hypoxia, simply that the patient will die of suffocation because the muscle tissue around the lungs will stop working. And then the last case, which is closer to what Gemma just spoke about, is disturbances of the blood's ability to coagulate and disruption of basal membranes. And this child has been bitten, I believe, by a Latin American viper that causes severe hemorrhage that both causes local effects, but which can also lead to more systemic effects. So back when before I sort of ventured into snakebite research, I was of course aware that getting bitten by a venomous snake was a very serious emergency, but I didn't expect that snakebite venoming was such a big problem on a global scale. So if we zoom out and don't just look at the individual victims, but look at the global numbers, snakebite envenoming causes somewhere around 2 million envenomings per year and leads to around 100,000 deaths. Snake bites, of course, occur mainly in tropical regions where there are a lot of venomous snakes, but you do find cases of severe snake bite on all continents. So you could say, fortunately, we've had antivenoms, and we've actually had them for more than 125 years. As Sakthi said in the beginning, the good thing about antivenoms is that they work. They are the only current treatment on the market, the only specific treatment against snakebite envenoming that will save the life of the patient. They are produced by immunizing a larger animal, typically a horse. And then once the horse has raised a strong immune response comprising antibodies, blood or plasma is collected from the animal, and antibodies from the plasma can be isolated and formulated into an antivenom. So this is a very robust and old technology that has been life-saving for a very long time. But with modern biotech approaches, what we, at least in my group, believe is that we have an opportunity to make antivenoms even better. And basically what we're doing is that we want to isolate genes of toxin targeting antibodies and express these antibodies in cell culture, in bioreactors, and see if we can make recombinant antivenoms that basically do the same thing as the existing antivenoms, but may have a number of benefits. So this figure shows how you could say, envision how the future antivenoms might be. I think that they will be comprised of a mixture between full IgG antibodies that have certain benefits, such as very long half-life, and which can mimic the antibodies that we know from horses. I also think it's likely that will include smaller antibody fragments that can have other benefits, such as fast tissue penetration. And I also think that some of the inhibitors also mentioned by Gemma in his talk could be very useful for targeting a number of particularly the enzymatic toxin families. So some of the benefits that we could hope that the future antivenoms might bring is that if you make the antibodies or antibody fragments in a human format and not an animal format, they will be safer, given that they are likely to be more compatible with the human immune system. They will simply not be recognized as foreign, as is the case with the existing antivenoms. Existing antivenoms also contain a mixture of all the antibodies that an animal has. So that means that it's only a subset of these antibodies that actually provide any therapeutic benefits in neutralizing toxins. If we go for recombinant expression and careful selection of the antibodies, we could avoid producing all the unnecessary antibodies. So you would have, say, more potent antivenoms, at least in a per gram basis. I think that one of the main benefits of moving away from animal-based production is also that there's an opportunity to make production more reproducible, to have less batch-to-batch variation. And I think this will also have a major positive impact on all the quality control practices that are currently in place, where we could save a lot of not just the production animals, but also mice in QC testing. And then I think the last thing that I want to mention is that I think if we can make antivenoms safer, I think there's an opportunity to deploy them much further out in the field if there's no risk of causing serum sickness and, even more importantly, anaphylaxis. So personally, I think we have a lot of opportunities that we should exploit in biotechnology to make snakebite and venoming treatment better. Generally, if we just focus on antibodies, there's two approaches that are currently being pursued in the field of antivenom research. There's the top-down approach, where you could say you start with an antibody repertoire from an immunized animal. So that's a polyclonal antibody response that contains both relevant and irrelevant antibodies. In principle, these antibody genes could be recloned into stable cell lines, such as CHO cell lines, and be expressed so that it would essentially be kind of a biosimilar to an existing antivenom. The benefit of that is that one could start from an existing production animal where we know the antibodies work and we know that the antivenom is effective. The downside is that you will either need to weed out a lot of the irrelevant antibody genes or perform a fairly complex selection or screening procedure to only include the relevant antibodies. The alternative approach, the bottom-up approach, is what we pursue in my lab. There we start with phage display, which I'll go into in a few more details in a little bit, where we basically take key toxins that are essential or very important to neutralize in case of human envenomings, and use these as targets to find specific monoclonal antibodies that can neutralize these toxins. And then you could say we go systematically through a number of venoms to find enough antibodies so that we can combine a panel up here that targets essentially all the medically relevant toxins. So that's the opposite way of building up an antivenom from the bottom, whereas the other one is a top-down approach where you start with more antibodies than you need and kind of move down. As mentioned, we use phage display technology in my lab to isolate human antibodies, or to discover human antibodies that target snake toxins. And here we make use of bacteriophages, which have the feature that they contain the antibody gene encoded in the DNA inside the phage. And this gene is inserted in a way so that the corresponding antibody fragment is displayed on one of the coat proteins on the outside of the bacteriophage. So this creates a link between phenotype and genotype. And basically what it allows us to do is that when you have a big library of a lot of different bacteriophages displaying antibodies, you can pan them in a small vial, where if your antibody library is diverse enough, you will very likely find some antibodies that will bind to the target, whereas all the antibodies that don't bind can be washed away together with the phage. We can then elute the phages, amplify the ones that bind, go for 1 or 2 more rounds of binding and elution to select the antibodies that bind most strongly to the targets. And then once we're satisfied, we will then analyze these different phages using ELISA techniques and DNA sequencing. So what one could compare the bacteriophage with is a big barcode, basically. So one could see the DNA as the barcode for the antibody, so that once you've selected your antibody, you basically scan the barcode by DNA sequencing and thereby get the primary structure of the antibody, so the amino acid sequence. One particular benefit of phage display in the field of toxinology is that many snake toxins are not very immunogenic, which doesn't matter in phage display. But when a toxin is not very immunogenic, it has a hard time raising a good antibody response in an animal, which is not such a big problem in phage display, because the ability of a toxin to raise an antibody response is not so important, because all of this is based on, you could say, very basic molecular interactions and basic 3D structure. So I'm going to take you through a few examples of our research, and I'm going to start with some of the work that I did, or at least started out doing in the lab on the black mamba. And here we took the venom, and I did this over at the Instituto Clodomiro Picaro under Chema's and Bruno Lamonte's supervision. But here we took black mamba venom and separated it into its different toxin fractions and identified which toxins were likely to be the medically most important in mammalian envenomings. So that means including humans. We identified 6 main toxin fractions that we evaluated needed to be neutralized. And these contain 2 different types of 3-finger toxins that are neurotoxic, and a number of dendrotoxins that are special other types of neurotoxins found in mamba venoms. We then proceeded to do phage display in Iontis' laboratories under the guidance of Anish Kirat-Velat and John McCafferty. And simply by taking the toxins as targets and panning their antibody libraries against the toxins, we could amplify phages that bound the toxins we wanted, but didn't bind other targets. What we do in an antibody discovery campaign is that once we've accumulated a polyclonal pool of antibodies that bind, we then reclone the antibody genes and express them in a soluble format and test them by monoclonal ELISA, as you can see over here. Our antibodies contain a tag so that we can detect if we have expressed antibodies that bind to the target. Once we've selected our hits, we proceed to do DNA sequencing to find out how many of the hits are unique. And we evaluate this looking both at the entire sequence, but also by digging more deeply into differences in the heavy chain CDR3 region, which is typically the dominating region that is the main driver for strong binding. We then reformat our single chain antibody fragments into full human IgG antibodies that we then retest to see if they retain binding by a different type of ELISA. And to go into the more interesting thing we did in this project, which was then again done at Instituto Clodomiro Picaro, we took 2 different cocktails, one cocktail comprising 4 human antibodies and another cocktail just comprising 3. And then we tested them against whole venom in mice that had been challenged with the black mamba venom. And at different doses, what we could see was that both cocktails were able to protect mice when they were injected cerebroventricularly with venom. I think it's important to say here that this experiment was done by pre-incubating venom and antivenom or experimental antivenom. So this was not a rescue experiment, which is a thing we would like to do in the future. So this example shows how mixtures of monoclonal antibodies can, to an extent, mimic what is seen with existing antivenoms and neutralize the effects of certain toxins in whole venom. Moving beyond this particular project and into a different one, looking at spitting cobra venoms, my postdoc, Shirin Amadi, did a very nice experiment where she took cytotoxins from different African cobras. You can see the different venoms here, And I've pointed out some of the cytotoxins that we've been working with. And what she did was that she did a so-called cross-panning experiment using the phage display technology. And in this experiment, she, in round 1, panned all her antibodies against toxin A. Then she took the subset of antibody binding, phages that had antibodies that bound. She took the subset of those and then panned them against toxin B to see, out of all the antibodies that bound A, which ones would also bind B, so that she would drive the selection towards cross-reactive antibodies. She followed many of the same procedures by first detecting, did she accumulate binders in a polyclonal ELISA format. Then she recloned all the genes into new vectors and did transformations that expressed the single chain variable fragments and retested them, and fortunately found many hits where she could see the individual fragments, at least at this point, bound 2 different cytotoxins. After some sequencing and identification of the unique antibodies, we recloned some of these single chain variable fragments into, again, the human IgG format and ensured that they retain binding. Again, the more interesting experiment here was that she took keratinocytes, that you can see over here, human keratinocytes, in a cellular assay, and both treated the keratinocytes with different toxins and venoms and observed the behavior. And what you can see here is that basically the cells die and they kind of clump together. And then she also tried to subject the keratinocytes to venom and toxins in the presence of some of the different antibodies that she found. And what she was able to demonstrate was that these antibodies that had been specifically selected to be broadly neutralizing could neutralize not just different cytotoxins from the same venom, but could actually neutralize cytotoxins across all 3 African cobra venoms. Moving even more, you could say, back or into Costa Rica, and a final experiment that I want to talk about is focusing on myotoxins from the snake Bothrops asper or Terciopelo, a viper that causes a lot of severe bites in Latin America, especially in Mesoamerica. And here I have a PhD student, Christopher, who has been working for a while on identifying, again, human antibodies that can neutralize these myotoxins that induce muscle damage. This hasn't been published yet, this work, but what Chris has done is a little bit similar to what you saw before. Used a different test to demonstrate that he's accumulated bacteriophages that have antibodies that bind the toxin. He's recloned the antibodies, tested which ones retain binding when they're expressed in a soluble format. He did a lot of sequencing and identified a lot of unique clones and have now reformatted them and tested that they retain binding. Again, the more interesting experiments that have been done is that Chrissy both tested the ability of one of his antibodies to neutralize myotoxin 2 from the snake in the same assay as Sheeran used, and could see that the antibody at different ratios provided full protection, at least in the higher doses. And then some ongoing work that is being carried out by Dr. Lemonte from ICP is that we've tested our antibody in mice that were challenged with myotoxin. And these are very preliminary results where we haven't really gotten enough data to make firm conclusions. But what it looks like is that the antibody does work against the toxin, but we need to optimize some formulation, as we saw certain increases in creatine kinase in the background of the formulation. So basically, what we believe is that the antibody seems to work in vivo, but we need to document that a bit more firmly. So now I want to move a bit away from our wet lab experiments and talk just a little bit about development and production, which I think is a very important topic when you talk about future envenoming therapies or future therapies for any indication in general. And here it's going to be a bit more conceptual. And the first thing that I want to sort of highlight is that one of the benefits of going into recombinant antivenoms is not that it's super cheap to develop. It's much more expensive to develop than going into traditional antivenom manufacture. But the benefit is more long-term on the manufacturing cost, where it seems when you look at how cost of manufacturing within monoclonal antibodies is developing, that there will likely be a chance to manufacture recombinant antivenoms in bioreactors at a lower cost than what immunization might be able to provide. So I think that's important to split up development and manufacturing, and when you evaluate the feasibility of moving into something. So one has to decide, is the initial R&D cost, that investment, is that worth the long-term potential gain? Another thing that I also want to talk about just conceptually is, you could say, what type of formats, what type of molecules are relevant to bring into the clinic. And I've seen certain papers, if I have to criticize, where people suggest quick fixes. And on the right side, you see my home and where my department is, and you see it's a very, very short walk. And here, a quick fix could be an electronic scooter or a bike or even walking. But developing a completely novel antivenom is not a quick fix. You cannot just take a molecule that shows a little bit of promise and then start using it. It's more like a trip from Copenhagen to Tasmania, which you would never obviously do with a small and simple molecule. So my point here, which I hope gets across, is that one step forward in developing a novel antivenom is really irrelevant if that step is in the wrong direction. You have to take the entire trip into account and only choose processes, technologies, molecules that can, you could say, endure the entire journey. And I'll come back to some of the examples of molecules which I think can, and some which I think will have challenges. It also becomes important at looking at what are the different antibody manufacturing strategies that can be used. And I'm not going to go into too many details here, but I'm just going to highlight a paper that I wrote recently with my postdoc, Timothy, where we tried to look at what molecules, what processes are likely to be economically feasible to use for manufacturing recombinant antivenoms in the future. And I've just highlighted a few takeaways. And basically, I want to highlight one important thing that people, they typically neglect. And that is the cost of manufacturing of a product is not only the cost of the API, the active pharmaceutical ingredient. If the active pharmaceutical ingredient is very expensive, the cost of fill finish or formulating and bottling is very low compared to the rest of the price. But if you work with very cheap active pharmaceutical ingredients that are dirt cheap, then the cost of fill finish, putting it in, formulating it and putting it in a vial is actually the main cost driver. So I think that's something that's worth taking into account, that API cost is not everything. On the right-hand side, I have sort of a simple table where we estimate, based on a number of different assumptions, what it will cost to neutralize a venom depending on how many toxins, so basically the venom yield. So is it a big bite or a small bite? And also depending a little bit on how big are the toxins. And the reason why this is important to take into account is basically that neutralizing a venom is kind of a numbers game. The more toxins you need to neutralize, the more antibodies you need. The more antibodies you need, the more grams of antibody you need to produce, and the more expensive it becomes. So this is why it will be a lot more expensive to neutralize a bite from a very big cobra that might be able to deliver many hundreds of milligrams of venom, compared to a very venom with a very low average toxin molar mass, compared to a viper with a smaller bite that has very big toxins. So I think it's important just to highlight that this has a dramatic impact on the estimated cost of manufacture for the API needed to neutralize a bite. We tried to estimate in this paper what will it cost to create a recombinant polyvalent antivenom against I think it's 10 most important snake species in Africa. We kind of tried to see what would it cost to make a recombinant biosimilar of Vaiyapuri, a very successful antivenom. And as you will see here, another important thing to take into account is the molar ratio needed has a very big effect on the cost of manufacturing. And the takeaway message is basically we need very good antibodies so that very few antibodies, preferably just one per toxin, is needed. Whereas the degree of cross-reactivity also has an effect, but actually has a smaller effect than how good the antibodies are individually. The final thing I want to highlight is that beyond the classical format of immunoglobulins, which are very big molecules, expensive to produce, there might be a chance, or there are opportunities to move into simpler, smaller formats, where less material is simply needed to neutralize a certain number of toxins. And again, don't take these numbers as precise numbers. These are estimates that are just meant to show that there might be opportunities of moving into alternative scaffolds, such as just Fabs, or maybe things like single domain antibodies and DARPins and other types of molecules. My final slide is something that I might save a little bit for some of the questions and the discussion, but what I've tried to highlight here is that there are a number of different proposed molecules that might be sort of the basis for developing novel antivenoms. What I personally evaluate or assess is that I think that recombinant antibodies and nanobodies, they are very promising, as well as repurposed pharmaceuticals. Gemma highlighted batimastat, but people at the Liverpool School of Tropical Medicine, I think I saw Laura here, she was at least here earlier today, have done some amazing work using both repurposed pharmaceuticals and small molecule inhibitors and chelators. I think those are very relevant to consider including in novel antivenoms, because you save or not the entire, but you save a big part of the research and development process and can kind of lower the R&D investment, as you saw in one of my slides, to move into the clinic. Some of the types of molecules that I don't think are that interesting, despite what might be highlighted in certain review papers and some research papers, are peptides and DNA and RNA aptamers. The reason I don't see that they have a huge potential is that they might be very versatile molecules, but there are a lot of inherent stability issues that need to be overcome compared to antibodies and nanobodies, which are scaffold molecules of known stability in vivo. And I simply think that it will take too much effort to sit and optimize every single peptide and aptamer for it to be feasible to make a big mixture of these type of molecules to neutralize a venom. I hope someone will prove me wrong about this, but that's my assessment, that the cost of development and the cost of manufacture for these type of molecules is simply too high, because you need to modify them too much compared to what you need to do with an antibody that we already know works. I think in between where we are now and maybe a future where fully recombinant antivenoms might exist, we might have an overlapping period where immunization strategies can be optimized and can provide more potent and more broadly neutralizing antivenoms. And I think some exceptionally interesting work has been done by Guillermo de la Rosa, also together with Instituto Clodomiro Picaro, where they've shown that recombinant immunogens, so recombinant toxins that were specifically designed, could be used to raise more broadly neutralizing antibody responses. So I think I'll leave it at that, and then I look forward to be challenged on some of these things in the questions. And with that, I just want to thank a lot of collaborators. I think I might have forgotten you, Sakthi, but you should actually be on there. But so far, I only included people that I've published with, and that is actually incorrect because we have published together already. I need to update this, but I want to thank a lot of different, both young and senior scientists that have been absolutely invaluable in getting some of the different results I showed here done. So thank you all for your attention, and I look forward to answering questions.
Thank you so much, Andreas, for this such a brilliant talk. So we got a few questions. Before I go through, I want to kickstart with my questions. Could you please kickstart on heat stability of these recombinant antibodies? Do you think we can easily store them in local healthcare settings, because that's one big problem we are facing with the current antivenoms.
Yeah, so that's a good question, Sakthi. And I think it's also something that should be taken into account when selecting what format of antibody you want to work with. IgGs, human IgGs, are neither more or less stable than horse IgGs. The same goes for the standard fragments such as Fab and Fab2. So I think the question is more, do we really need this improved stability? I mean, a lot of papers say so, a lot of people argue so, and I'm not necessarily of a different opinion, but I'm just saying you can get cold Coca-Cola in pretty much any part of the world. There are improved lyophilization techniques that can extend half-life. No doubt that it's better to have heat stability, but is it more important to get safer products and effective products than it is to get more stable products? That's one way of looking at it. That being said, if we move into things like single domain antibodies and nanobodies, it's well known that these type of molecules are very stable compared to the classical immunoglobulins. In a different project in infectious diseases, I've been working with nanobodies that can be pasteurized and still retain their binding strength. So meaning that they're still stable. So no doubt that there's opportunities, but I think it's important to consider what is the most urgent need right now. I'm not sure it's stability when we talk about novel antivenoms. It might be important for existing products to improve stability, but I think it's something that definitely is worth investigating, but maybe not the first thing we should look at improving.
Yeah. So we got a few questions here in the chat box. We can try to answer a few. So how consistency of produced forge are checked, and how many monoclonal forge you usually choose? I think you covered some of this. Do you want to touch on it?
Yeah, I can say that in every selection campaign, we use about 10 billion antibodies, if we go with a universal library. We are also using nanobodies, and in those cases, we sometimes come with immune libraries where the diversity is much smaller. But these antibody libraries are based on immunized llama antibodies. So we don't need that big repertoire.
So do the forge antibodies repeatedly recognize the conserved sequences of the toxins, which make them broad spectrum antivenom?
That's a super good question as well. It's not something we've investigated that much. So we are trying to the extent possible to find antibodies that recognize conserved regions, and theoretically using the technique of cross-panning, where you have two different toxins that might share one conserved region. The only way an antibody will be successfully selected is if it binds that particular region. It would simply be lost if it didn't. So I think that the answer is likely yes, but it's not something I can say that we have documented.
Okay, one last question. Which format of the antibodies, from your experience, either single chain or single domain or Fab fragment or full antibodies, has the most optimum pharmacokinetics properties for administration? Yeah,
I personally think, at least for how the world looks now, it will be a mix. I mean, it's both immunoglobulins, human IgGs that have a long half-life that when injected IV will circulate for many weeks and protect against recurrent envenoming from toxins leaking from the bite site. I think that format is very beneficial pharmacokinetically against systemically acting toxins, especially like neurotoxins, where you just need to intercept the toxin before it reaches its target. But I also think that smaller molecules like nanobodies, which have by a Belgian group been demonstrated that they really can penetrate into deep tissue much faster than larger antibodies. They also have some very interesting pharmacokinetic properties, particularly for locally acting toxins, where it may be important to get out quickly and start neutralizing the venom. So I don't think there's one format that is just universally better than any other, but there are some formats that are beneficial against certain subfamilies of toxins, as I see it.
Excellent, thanks, Andreas. There are a few more questions. It will be great if you can answer them in the chat box, because of the limited amount of time, I will kickstart. Thank you, Andreas. Okay, so the next talk is from me. So I'm going to talk about the community health education. So let me start sharing my screen. So as I said at the beginning, so despite we do all the research and the developments for developing new diagnostics and the treatments, it's critical that we develop appropriate awareness among people locally. So they are like mostly rural communities who are the vulnerable people for snakebite. And at the same time, we need to focus on people who are handling snakebite victims in the rural settings as well, mostly rural healthcare professionals, including clinicians, nurses, pharmacists, these type of people, because they are the one going to handle the snakebite victims on the ground. So that's also important. And then the third thing, we need to talk to appropriate policymakers. They are mostly local governments, and we need to convince them in order to update the current policies and develop new policies as necessary. Therefore, as researchers in this field of snakebites, we do have a responsibility to develop appropriate awareness among people. So I'm going to talk about today about the snakebite awareness program that we've been running successfully for the last 2 years in Tamil Nadu in India, in South India. So the background for snakebite, we all know about snakebites. So snakebites mainly affect the rural population, and it's a predominant problem. And most importantly, the people who are affected by snakebites are agricultural poor farmers. And these people are struggling to earn small amount of money for their everyday life. When a snake bites them, it can completely change their life upside down. And there are lots of perception and myths about snake bites still in rural communities. We might think that this is a modern world and fully educated and people are aware of everything, people do sensible things, but it's not the case. There are so many perceptions, so many myths, misbeliefs available about snake bites in rural communities, which are ultimately affecting their treatment-seeking behavior. And the other thing is influence of traditional medicine and inappropriate first aids. So there are traditional healers mostly available locally, and they are cheap, and in most cases they are free as well. So therefore, as soon as snake bites someone, immediately people will try to seek treatment from these local traditional healers, which is significantly delaying their hospital treatment. And also inappropriate first aid. There are so many inappropriate first aids because these have been disseminated via movies, documentaries and other books, this and that. So people have this implanted in their minds. So still, if somebody is bitten by a snake, immediately they will take a knife or anything, it could be contaminated, immediately cut the area, try to suck the blood, and put tight tourniquets, which is going to affect quite a lot down the stream when they go to hospital. And the other thing is lack of adequate medical and transport facilities. So if you go to countries like India, there are thousands of tribal villages still available, and these people still don't have electricity, road facilities, and therefore there's no transport facilities. So if they need to come for their essential things, they need to usually walk for several kilometers. So the same thing for medical facilities as well. So they don't have adequate transport and medical facilities to seek immediate treatment. But if you look at the healthcare system in India, so for every 10 to 15 kilometers or maximum of 20 kilometers, there will be a small primary health center in the local healthcare setting. But do they have enough expertise and facilities to handle snake bites? That's a big question that we all need to ask. So most of these are primary health centers, and they can handle minor illnesses. Well, when it comes to snake bite, it's a complicated problem, okay? It could be a dry bite or non-venomous bite where we don't have to worry about much about the complications. But in cases of severe envenomation effects, it's going to be multiple complications because snake venom can affect all over the body. So in these cases, there's a lack of expertise and facilities in local healthcare settings, and therefore these people are not confident enough to treat snakebite victims. Therefore, they keep on referring them to tertiary medical care, which is taking a long time for these people to reach. And the other thing is there's always limited attention from healthcare authorities, particularly the current pandemic, you all know. So COVID-19 has taken over all the priorities from all healthcare authorities. Similarly, in other cases like HIV, malaria, hepatitis, these things always take the priority compared to snake bites. because healthcare authorities always think that there's nothing more to do for snakebite because they are providing antivenom in subsidized rate and it's all available. And snakebite is not a big issue because there's no reliable data. So we got no idea how many people are being affected by snake bites. So all of these factors are leading to snakebite induced complications. It is exacerbating the complication in several cases, which is ultimately leading to increased number of deaths, disabilities, and most importantly, socioeconomic impacts. As I mentioned at the beginning, a single snake bite can change somebody's life or their family's life completely upside down. So the most important thing is lack of reliable data. So recently, there are several papers being published about the actual statistics about snake bites, but still there's a huge underestimation of that number because many people don't go to hospital following snake bites, and many people die on the way to hospitals, and many people in countries like India seek treatment from private hospitals where the health statistics is not being collected. So therefore, together, worldwide, there's a huge underestimation on the burden of snake bites among the rural communities. So we did two studies back in 2010. So the first study we did was household survey and interviewing snakebite victims. So we all knew that the snakebite number has been always underestimated. So we planned a study to do in rural Tamil Nadu. So we wanted to do a household survey to determine the actual number of snake bites. It's not going to be accurate because it was a pilot study, but at least it will give a hint on how many snake bite cases are occurring and how many people are dying. So we did a household survey in 30 villages randomly chosen in Tamil Nadu, and we estimated the number of snake bite incidences and deaths for the last 10 years. And that gave a really shocking number. So compared to Millennium Death Study, which was published along when we were doing this study, that reported around 3,000 incidences, 3,000 deaths in Tamil Nadu. But this study found out that in Tamil Nadu alone, there's likely to be 10,000 deaths every year. And whenever they have heavy rainfall, the death number can go up. And most importantly, we interviewed 105 snakebite victims who were recently affected by snake bites and determined various factors around the snake bites, how this is affecting or how this is causing socioeconomic impact. So I hope you can see this table. So I want you to have a quick attention here. So while there are no first aids in 83 cases, still many people are practicing other inappropriate first aids. For example, some people are using tourniquets. Most importantly, some people put like multiple tourniquets above the bite site. And some people are using tourniquet and incision. They will cut the bite site using non-sterile blade or knife and then suck the blood. And some people apply calcium carbonate because they think it can cure or it can neutralize the venoms. And some people apply toxic secretion from plants, for example, calotropis, and then some people force them to vomit. So these are the things are happening. And then as I said before, in countries like India, many people try to seek medical attention only from private hospitals, even though the government hospitals are free and they provide a range of different treatments. People always try to seek treatments from private hospitals for various reasons. So in some cases, there's no treatment cost because it could be a non-venomous bite, or it could be a dry bite, or it could have been completely treated by a government hospital. But in other cases, the treatment cost could vary from 1 rupee up to 3.5 lakhs. That's 350,000 rupees. It's around 5,000 pounds. But this was back in 2010. So the treatment cost is a substantial factor here. Okay, so in some cases it's really small, it can be manageable, but in some cases it's huge amount of money for poor agricultural farmers. And then if you look at this area for further treatment, many people seek treatment only from hospitals, but some people seek treatment only from traditional healers, and some people treat initially from traditional healers, and then when the situation gets really worse, then they will go to hospital, and that's where it's becoming a challenge to treat these people. Because as Chema has explained, the local envenomation effects will be hugely developing, and that's when the clinicians have to go for surgeries or amputations. And in many cases, people have to just receive antivenoms. And then the most important here is because people don't have a sufficient amount of money to pay for their treatment, and they are not covered by any health insurances. But these days they do have a health insurance, but when this study was done, there was no health insurance. So therefore, people have to sell their valuables to pay for treatment, and some people have to take loans, private loans, because there's no loan available from nationalized banks. And most importantly, some people had to sell their land or house, and some people had to remove their children from education in order to send them to work to earn some money to pay off their loan. So the key outcomes of this study was there's a basic lack of awareness about snakes and snake bites among rural population. And in several cases, people are traveling such a long distance to reach tertiary care hospitals without any proper first aid or without giving even 2-3 vials of antivenom. So the delayed hospital treatment is the biggest problem. So if people reach hospital within like half an hour or 1 hour, they can be treated very quickly. But when they reach after 24 hours, 48 hours, it's becoming a really big challenge because of exacerbated complications. And in these cases, the treatment costs will be obviously very high because of the surgery and other medications they have to give, dialysis, lots of other things. It's all substantially increasing the treatment cost. And as I mentioned, there was no health insurance before, but there is a health insurance at the moment in some states, particularly in Tamil Nadu, but still it doesn't cover all the cost, which we can talk about later on. And then at the same time, when we did the study, we interviewed some clinicians to understand what are the challenges they are facing in diagnosing and treating snake bites in rural settings in Tamil Nadu. So here we found out several things. First thing is not everyone is confident in treating snake bites. Even though there are private healthcare settings, government healthcare settings available everywhere, not everyone is feeling confident to treat snake bites because of the multitude of envenomation effects we are getting with the snake bites. So that's a big, big problem. Therefore, only well-experienced clinicians are treating snake bites, and they are located only in limited places. So that's why these people have to travel such a long time to reach these people. And then even though there's a WHO standard guidelines and government guidelines from Government of India available for snakebite envenomation, not many people are using these standard protocols. They mainly rely on their own expertise and experience. So that's how they are treating snake bites. And other people don't have access to these protocols. That's also another problem. And the main challenge they also highlighted is delay in seeking treatment. Okay, they feel really confident in treating snakebite victims if they arrive promptly to the hospital without practicing any inappropriate first aids or without seeking any traditional treatments. And in some hospitals, still this is a big problem. There's a lack of adequate supply of antivenom. Okay, because in India, I think there are only 3 or 4 suppliers for manufacturers for antivenom, and there's a demand for antivenom all the time. And the treatment cost could be very high due to multiple factors. As I mentioned before, it could be because of dialysis, surgeries, other things. It's costing more money to treat these patients. And then last important thing is lack of awareness among healthcare professionals who are particularly working in rural settings. Okay, these people are like less experienced junior healthcare professionals, and they never heard about snake bites, and therefore they don't have a confidence in treating these people. Therefore, they are making the victims to travel such a long time, which is causing a lot of problems. So in 2019, or in precisely 2018 December, we launched a mega public awareness campaign in rural Tamil Nadu in collaboration with multiple people, which I will talk about in a minute as well. The main aim of this snakebite public awareness campaign is to develop awareness about snakes and snake bites among rural communities in Tamil Nadu. And at the same time, we wanted to raise awareness about snakebite management among healthcare professionals. These two are the critical aims, and then finally, we will influence the policy makers based on these outcomes. So the study design, this study was mainly designed to do it in rural Tamil Nadu. Tamil Nadu has got a 77 million population, and around 35 million people are living in rural areas in the state. And we aim to reach around 10% of this rural population within like 1 year, and we reached that population. And we developed specific campaign tools because it's not always easy to just to go and give a lecture and then walk away, and that doesn't work. So you need to develop various materials. So we developed various materials, which I will explain in a minute. And then we deployed a multifaceted approach. Okay, only through direct activities, there's no way we can reach large number of people. Therefore, we used direct activities in schools, colleges, universities, and other public places. And we utilized the support of media in India, in Tamil Nadu, and also widely in India, because that's really important for this type of activities. And we also used social media for this study. Then the next thing is evaluating the impact activities. We can be doing hundreds of impact activities everywhere, but we got no idea whether it's impacting on the people or not. So we came up with like unique approaches to evaluate the impact activities that we were doing. And then we developed a symposium for healthcare professionals where we gathered nearly 250 healthcare professionals and talked about various aspects. And currently we are running an online survey to determine the knowledge gaps and training requirements for healthcare professionals in order to develop appropriate materials to train these people. So what are the campaign tools we developed? We developed snakebite information leaflets with basic information that people can give these, we can give these leaflets to people, and people can keep these leaflets in their house to look for this information whenever necessary. And we developed some information posters that we can stick in public places and hospitals and primary health centers. And then most importantly, we developed lots of documentaries, long and short documentaries with key messages about snake bites. And we also ran like specific workshops in some educational institutes. And we developed a dedicated Facebook page for this activity. And we developed evaluation questionnaires for rural communities and also monitor the SPE victims admission in a referral hospital in India. And then finally, we developed a comprehensive online survey for healthcare professionals in India. So this is the information leaflet to quickly show you what sort of information we are providing. Okay, so these are all printed as hard copy. We disseminated over 200,000 leaflets so far. We are printing another 200,000 now. So this is giving sort of like a key points about what to do if a snake bites you, and what sort of first aid you need to do if a snake bites you, and how to avoid snake bites around your living areas, and where to call for help, in case of emergency. And also we are providing our collaborating doctor's name here from TCR Multi-Specialty Hospital and their phone numbers, so they can also seek support from them. And then on the other page, we are showing the big four snakes, because many people are not aware of big four snakes, which again, I will talk about that later on. And these are the big four snakes causing most of the problems in India. So we are talking about the clinical symptoms of these snake bites, and also rapid action. So what you need to do, what you shouldn't do if somebody is bitten by a snake immediately. So this is quite important, and this leaflet formed a really critical part during this campaign. And as I said, we distributed this leaflet to over 200,000 people. And the next thing, we printed this type of posters, both in Tamil and in English. And this has got only key messages like how to avoid snake bites, or what to do if a snake bites you, and then the clinical symptoms of the big four snakes. So these posters we stick in hospitals and other public places, and we are distributing to for many people as well to stick in their places. And then in collaboration with the Health Action International from Netherlands, we adapted their poster, which they have developed for Sub-Saharan Africa. And then here they highlighted clearly the do's and don'ts for snake bites, multiple actions using these beautiful cartoons. And this was really effective in African countries. So we adapted these, we added Tamil translation here. And now we printed these posters and we are disseminating this many places, and we are also disseminating these posters via online as well. So people can use these type of things to quickly see what they should do, what they shouldn't do if somebody is bitten by a snake. And then we developed one big documentary, again in collaboration with multiple people like Dr. Stephen Paul and Dr. Soundarraj from TCR Hospital, and some snakebite victims, rural people, and other researchers who are researching on snakebite, and some snake rescuers. So we developed a 25-minute long documentary with all the key messages that we can tell to people about snake bites. And this also played a critical role during our campaign activities. And as I said, we developed a dedicated Facebook page, and where we were just disseminating all the materials that we developed for this campaign, and we disseminate about our activities we are doing in different places in Tamil Nadu, and also we disseminated lots of media articles that were published during this campaign program as well. And most importantly, this page provided a platform for people to ask questions. People are asking multiple questions about snakes and snake bites through this page. For evaluation, after our activities, we wanted to find out whether it really impacted, whether people have increased their knowledge about snake bites following our activities. So we adapted Health Action International posters, and we adapted those cartoons and then came up with like specific actions. And then we give this questionnaire to people in schools, colleges, wherever it is, before and after our activities. So before activities, we can gauge exactly how much knowledge they have about snakes and snake bites. And then immediately after our activities, we will give this questionnaire and ask them to fill in the correct answers. Then we can find out how much knowledge they got increased. Or also we did it after 1 year to check their long-term memory about the impact of these activities. So working team, this type of work is impossible to do without sufficient amount of personnel. And this is our TCR multi-speciality hospital. It's a snakebite referral hospital in North Tamil Nadu. And Dr. Saundarraj and Dr. Stephen Fall from this area, they are the critical collaborators for this study. And we also collaborated with numerous other hospitals, pharmacies, and NGOs. One of the team is like here, Urvanam team. They cover lots of activities for us in South Tamil Nadu from Madurai and downwards. And we also worked with the snake rescuers, lots of journalists and media partners, police, forest officials, and village public health officers. So we started this work with a team of like 10, 15 people. Now we do have more than 100 people working for us at different stages in Tamil Nadu for these activities. So we performed several activities. So for example, in educational institutes, so we covered more than 140 educational institutes within a year. So we approach this educational institute like schools, colleges, universities, and then arrange a specific time to arrange a snakebite assemblies. So where we will go along with the clinicians and some scientists, snake rescuers, we will go and we will give a small public lecture and then play a documentary about snake bites. And then we will run a massive question and answer session where students can ask any questions. We will answer them all. And then finally, we distribute our leaflets and then evaluate the knowledge they developed as well. Similarly, we go to rural villages in all the regions and we distribute the leaflets and give a small speech about the snake bites and answer all their questions. And if it's the evening time, we can also play our video documentary in community centers or common places like markets or bus stations, and then try to answer all their questions. So this has been exceptionally effective. And you can see here, this is Tamil Nadu. I don't know whether you can see the boundaries, but this state is Tamil Nadu, this side is Kerala. So we covered activities in more than 100 different locations in this state. And we covered more than 150 institutions and more than 200 public places. And as I said, we got a massive network support at the moment. So multiple people across the state who are distributing our leaflets, and they are continuing to run this type of snakebite assemblies in multiple places. And physically or directly, we reached over 250,000 people so far in schools, colleges, and villages. And using social media, we reached over 3 million people. And then most importantly, using media, we published over 100 articles. And through these articles, according to the readership statistics, we could have reached more than 70 million people in the state and across the country as well. So because we used a multifaceted approach, it's been very effective to reach a large number of people within a short time. So as I said, media support was significant. Okay, so everyone, when we approached them, they understood the significance of this problem, and therefore they published far too many articles. Over 100 articles were published so far about our campaign, both in Tamil and English, and some of them local to Tamil Nadu, some of them across India. So all these articles disseminated key messages about snake bites and what to do, what not to do if somebody is bitten by a snake. So this has been a very critical for our campaign. And then Facebook, as I said, we got more than 20,000 followers here. In Tamil Nadu alone, for example, we got 18 million Facebook users in Tamil Nadu alone. So we targeted several activities, specifically for Tamil Nadu, and we got over 20,000 followers. We disseminated lots of materials, activities, and media articles. And most importantly, we used Facebook promotion option to disseminate specific videos. So through these type of activities, we engaged with over 3 million people through Facebook. And then, as I mentioned before, we ran a healthcare symposium on snake bites. This is exclusively for healthcare professionals. So we invited lots of rural healthcare professionals. We received over 250 attendees, and we covered a range of different clinical aspects about snake bites here. And it formed a network of clinicians, and still some of the clinicians are asking us or contacting us for clarifications, or when they are handling snake bite victims. This has created a positive impact for rural healthcare workers, because many of them have never heard about snake bites or never thought about managing snake bites. But now a couple of hospitals are managing snake bites just based on this symposium. And we identified the need for more training. Everyone at the end of the day asked for more hands-on training, more materials in order to manage snake bite victims in their local settings. So we evaluated the knowledge, because after doing all the activities, it is important to see whether these activities have caused any impact. So we, using the questionnaire, the pictorial questionnaire I mentioned, we evaluated the knowledge increase in schools and colleges. So here you can see the blue color ones, that's before activities. So for various questions, so only very few people, like around 40% answered correctly for some of the questions. For some questions, only like 10 to 20% answered correctly. Immediately after our activities, the knowledge was increased to up to 95%. And after 1 year, still that knowledge exists. Still more than 90% people remember all the key messages we ran, we provided during these assemblies. And then we also ran poster competitions in specific educational institutes, where students demonstrated the knowledge they grasped during our assemblies through posters. And this is how they disseminated how they will convert or disseminate the knowledge they gained from all their activities to their neighbors, friends, and other people around them. So this has been very effective. Many students highlighted the significance of seeking prompt treatment in hospitals following snakebite. So this has been effective. And similar activities we also performed in rural villages from villages. So they also highlighted increase in knowledge immediately our activities and also after 1 year. So this has demonstrated that definitely these activities using multiple approaches are effective to increase knowledge about snake bites among people. So that's a very good thing. The next thing we need to find out whether people have changed their treatment seeking behavior. They might have learned the knowledge, but they may not change their treatment-seeking behavior. They might continue to do what they were doing before. So we used TCR Hospital as a snakebite referral center and also to collect data. So we collected data from this hospital in 2018 when we didn't do any awareness activities, and then data in 2019. So we compared this data and see whether any differences had. So you can see here the snakebite total cases were increased in 2019 compared to 2019. Age-wise distribution, we didn't see much difference because this is a trend we always see because snake bites mainly affecting the working age growth. But when we come to the time taken to reach this hospital, we saw a significant difference compared to 2018. In 2018, you can see, even though a large number of people coming in 0 to 8 hours, there are still a lot of people coming up to even 48 hours. But in 2019, majority of people, like more than 90% people arrived at the hospital within 4 hours, which is really dramatic change. So people are trying to seek hospital treatment as quickly as possible following these activities, and that's very good. And then if you look at the type of first aids they are taking, the significantly higher people came without any first aid, which is a very good thing. And some people started using the pressure bandages because previously people are using only tight tourniquets. That has gone down in 2019, but notably people have started using pressure bandages instead of using any kind of rope. They started using this type of elastic pressure bandages, which is also a dramatic change. So these activities indeed changed the treatment seeking behavior, and we collected data from only one hospital, even though we performed activities across the state. So these type of things can be useful to evaluate the impact of our activities you are doing. Then the next thing quickly I want to touch on is an online survey for healthcare professionals. So based on our symposium, we developed this survey to determine the knowledge gaps and training requirements across healthcare professionals across India. So basically we developed, my PhD student Anika Salim has developed this using a comprehensive survey. Everything is online, everything has been validated. So this is basically capturing multiple level of knowledge from people, like background details, their qualification, gender, age, education, and then place and nature of work, and their general knowledge about snakes, particularly big four snakes, whether people have heard about big four snakes, whether they can confidently identify big four snakes, and the knowledge on preventive measures, appropriate first aids, education and training on management, and then snakebite management training requirements, and then if they treat snake bites, their level of expertise on clinical symptoms, antivenom, specific procedures. So this survey is asking multiple aspects from people. So far we received responses from 375 people in India. You can see lots of males and females, and many people are in the main age group. And the majority of responses, even though it came from Tamil Nadu, we did receive lots of responses from other states as well. And many of them are doctors, and some of them are medical officers, nurses, pharmacists, and other professions. We also captured different educational qualifications. And then many people said that nearly 41% people said they are aware of the venomous snakes and non-venomous snakes available in their region. But nearly 40% people said they know partially the important snakes. Most importantly, they all know about the cobra. And several people said they are not sure about the snakes living in their region. And then when we asked about big four snakes, around 59% people heard about big four snakes, but nearly 41% people never heard of big four snakes. And then when we asked them to identify the big four snakes, as expected, most of these people have identified cobra because of the hoodie behavior, but many people struggle to identify the remaining snake, particularly for saw-scaled viper. And then knowledge on first aid, again, many people don't have knowledge about the snake bite, appropriate first aid for snake bites. As you can see, some people said incision and punctures, sucking blood would be appropriate first aid for snake bites. So obviously this is highlighting the knowledge gaps among healthcare professionals. And here we asked about the knowledge they learned during their education, both theoretically and practically. Only around 48 or 47% people learned some knowledge during their medical education. And then we asked how many people feel confident to treat snake bites. Only around 50% people said we are confident to treat snake bites. And then when we asked whether they would be interested to treat snake bite if we provide appropriate training, nearly 97% people said yes, we would be interested to handle snake bite victims if we are provided appropriate trainings. Then we asked lots of other things, like these are mainly from snake bite experts, people who are treating snake bites already, about the number of people they are treating in, and how often they snake bite victims, and then the type of diagnostic tests they are doing, and the determining factors to go for antivenom as well. So this study is almost completed. We are about to analyze and start the publication procedure, and based on the outcomes of this study, we will develop training materials for them. So in summary, the low-cost community health education program is really effective. This entire program so far costed only £25,000, but we reached nearly 3 million people, either directly or via social media, and using media support, we reached over 70 million people. So this type of study can be continued not only in India, also in other parts of the world where snakebite is a major concern. And we need to always apply multifaceted approaches to reach a large number of people. Physically, we cannot reach a large number of people because we are also human beings. There is a limit to how much people we can reach. And through these activities, we can easily break the myths about snakes and snake bites. And by implanting basic knowledge about snake bites on people, we can encourage them to seek appropriate treatment very quickly. And we can also develop online tools to determine the knowledge gaps and training requirements, not necessarily from healthcare professionals, but also from other stakeholders, including rural villages and other people, because mobile phones are being heavily used these days in countries like India. So we need to start to implement the mobile health approaches and other technologies as well. And then we can develop information and training tools, and that can be disseminated via multiple platforms. And these will provide confidence for rural healthcare professionals. If they have a confidence, as soon as they get a patient, immediately they can give some appropriate first aids, or at least they can give 2-3 vials of antivenom before they send them to tertiary hospitals. And then we can encourage villagers to seek prompt treatment. I know they are obsessive about traditional treatment or inappropriate first aids, but by keep on telling them the right things, we can encourage these people to seek prompt treatment. And then next step, we can engage with policymakers by collecting robust data, by collecting appropriate intervention data on their effect. We should be able to influence policymakers in order to adapt to the policies or change the policies, and they can also do these type of similar activities. So finally, I would like to thank all different collaborators, because as I said before, this work is impossible to do without multiple people. So we are collaborating with several hospitals and NGOs, pharmaceutical companies or biotech companies, and we received funding from GCRF and University of Reading for this work. And I would like to thank all of these people for supporting last 2 years to perform these activities. Thank you so much for your attention. I'm happy to take a few questions from you. So there's one question from Spiros Pachis. It would be interesting to see also the correlation between the age of the person bitten and how long it took to get to the hospital, and whether your campaign affected that. I assume younger people were impacted more due to the use of social media and technology in general. Could you say something about this? Yes, you're right. And that's why we are mainly targeting the educational institutes. But mainly these people, by giving this information for these people, we are advising them to go and disseminate this information for others, particularly their family members, their friends and relatives. And this is how we can disseminate the information. So I know they are gathering the information through these technologies, but it shouldn't be just for them. They should disseminate the knowledge for others as well. That's a good point. Okay, any other questions? Okay, if not, again, I would like to thank our lovely speakers for sharing their views and scientific expertise today in this conference. It's been a really successful conference with over 100 people. So I would like to thank MDPI Sci Forum for organizing this conference and giving us the wonderful opportunity to share our knowledge. And thank you so much for all your attention. Anna, do you want to highlight any specific points?
No, exactly the same. Thank you all the speakers for your excellent talks. Thank you all the participants of the session. As I already told you through the chat, tomorrow we will have the last live session of this Toxins conference. So just visit our webpage to know more about it. We will also post the recording of this session in about 2-3 days in the conference webpage. And this is all from my part. Thank you again to all of you.
Excellent. Thank you, everyone. Cheers.
Thank you.
Thank you. Bye-bye.
Thank you, Andreas. Thank you, Chuma.